Hello everyone, welcome back to my channel Anytime Medicine, and today we are going to learn about glycogen. So if you are new here, subscribe to my channel so you cannot miss my future videos. So the glycogen is the storage form of glucose, which is a polysaccharide in which there are repeating molecules of glucose. Glycogen most abundant found in muscles and liver, muscle used for their own and liver used for body to make enough energy. Here you can see the polysaccharide molecule of glycogen in which there are two main bonds. First bond is the alpha 1,4 glycosidic bond, which is the linear bond and which makes the chain longer. Second bond is the alpha 1,6 glycosidic bond, which is the branch bond and which makes branch in chains. Here you can see the whole process of glycogen synthesis. Glycogen synthesis occurs from glucose 6-phosphate. As you can see in the picture, that glucose 6-phosphate will convert into glucose 1-phosphate with the help of phosphoglucomutase. And glucose 1-phosphate will eventually make the glycogen, and branching enzyme will make the branch glycogen. Like we have seen before glycogen synthesis, now we are going to see glycogen breakdown, that how glycogen will break down into glucose. So first of all, branch glycogen will convert into unbranched glycogen with the help of debranching enzyme, and that unbranched glycogen will convert into glucose 1-phosphate with the help of glycogen phosphorylase. And from that, glucose 1-phosphate will convert into glucose 6-phosphate very easily, because this is the reversible process. And glucose 6-phosphate will convert into glucose, or that will go to glycolysis. If it convert into glucose, this process only occur in liver. There is another way that glycogen can be converted into glucose, in which unbranched glycogen will directly convert into glucose. This process will occur with the help of alpha 1,4 glucosidase, and this is only occur in lysosomes. Now we need to know about dextrins. What are dextrins? Dextrins are mixtures of polymers of D-glucose units, which is usually 2 or 4, linked by the alpha 1,4 or alpha 1,6 glycosidic bonds. As you can see that there are 2 or 4 molecules that are attaching with the alpha 1,6 and alpha 1,4 bonds, which is the small molecules of glycogen, as you can see. In glycogen breakdown, we need to know the two main enzymes. First one is the phosphorylase, which removes the glucose from glycogen polymer and make glucose 1-phosphate, which stops when molecule reaches to reduction. As we have seen before, that it is the 2 or 4 molecules of the glycogen, which stabilized by vitamin B6. And the another enzyme is debranching enzyme, which cleaves the reduction and make it even smaller. Before we saw the debranching enzyme, now we are going to see is what type of activity debranching enzyme does. First activity is the debranching enzyme transferase activity. As you can see in the picture, that these 3 glucose molecule will transfer into the linear branch of the glycogen. Second activity is the debranching enzyme glucosidase activity, in which there is this 1 molecule of glucose will detach from the glycogen linear chain. Now we'll see that how hormone can regulate the conversion of glycogen and the glucose. If there is high level of glucagon and epinephrine, it will convert the glycogen into the glucose. And if there is high level of insulin, it will convert glucose into the glycogen. There is another way in which glucose can convert into glycogen. It will depend on the phosphorylation and dephosphorylation of these two enzymes, which is glycogen phosphorylase and glycogen synthase, that we are going to see in the next slide. First we will see the enzyme phosphorylation, which favors the activity of glucagon and epinephrine, which increase the activity of glycogen phosphorylase and break down the glycogen, and decrease the activity of glycogen synthase and decrease the production of glycogen from glucose. Second activity is the enzyme dephosphorylation, which favors the activity of insulin, which increase the activity of glycogen synthase and make more glycogen from glucose. And it will decrease the activity of glycogen phosphorylase and will decrease the breakdown of glycogen into glucose. If we see those previous slides that we have discussed before into simplistic view, if there is high level of insulin, it will promote the protein phosphatase. If there is high level of protein phosphatase, it will dephosphorylate the glycogen synthase and it will promote the glycogen synthase enzyme. And protein phosphatase will phosphorylate the glycogen phosphorylase enzyme and it will decrease the activity of phosphorylase enzyme. It means that in this process there will be high level of glycogen. In the other hand, if there is high level of glucagon and epinephrine, it will promote the cyclic AMP production and it will promote the protein kinase A and phosphorylase kinase activity. Protein kinase A will phosphorylate the glycogen synthase enzyme and it will decrease the activity of glycogen synthase. And phosphorylase kinase will dephosphorylate the glycogen phosphorylase enzyme and it will promote the glycogen phosphorylase activity. So now we'll see how glycogen breakdown occur in muscle contraction. In muscles, calcium releases from endoplasmic reticulum and it will activate the calmodulin. Calmodulin will attach with the phosphorylase kinase and it will phosphorylate the phosphorylase enzyme. And from then, glycogen breakdown occur. We have seen that how hormone can regulate glycogen to glucose conversion, and now we are going to see that how certain molecules also can regulate the glycogen and glucose conversion. There is first one molecule which is glucose 6-phosphate will promote the activity of glucose into the glycogen, and there is another molecule which is AMP which promotes the activity of glycogen to the glucose. There is another that ATP and glucose will inhibit the process of glycogen to the glucose process. So now we'll see how glycogen can be used as fuel. After we eat, body will make energy from ingested glucose for several hours, and after that glycogen start breaking down and will make energy from glycogen breakdown. After that, body will start do the gluconeogenesis and will make energy from proteins and fatty acids. So thank you everyone for watching this video. If you like this video, hit the like button and subscribe my channel and share it with your friends.